All right. So day three, we'll be talking about active transport, and this will be the last day for the new information. All right. So active transport. So just like before, active transport is what we'll be talking about today. Requires energy. Remember that energy molecule is known as ATP. There are three types. The sodium potassium pump. We'll look at it: endocytosis and exocytosis. Mm. Make sure that remember, like I said on the day one stuff, you need to make sure that you understand the difference between passive and active. Make sure you also know which the types that go with their correct grouping. So do make sure you study that. So active transport, just like we have a concentration gradient for passive, we also have one for active. And what it is it's the opposite of it. It's the opposite of what was for passive. So the concentration gradients, whenever substances move from an area of low concentration to an area of high concentration, or we're going, a lot of times you'll see that saying that we're going against the gradient. Passive would use the term like we're going with or along the gradient, but active is against the gradient. It's kind of like if there's a rushing river trying to swim against the current. You're going to have to use a lot of energy, but I could sit there and put you in a tube and you don't have to use any energy to flow with the river. So that's the difference between passive and active on those type of terms. So right here to the right, I have an image, the opposite of what was on day one. So now we have this guy, and he's having to take this rock from the low side of that hill and push it up this hill. That's going to require a lot of work. That's going to require a lot of energy. That's moving from the low side, the low concentration, to the high concentration. Okay. So this is an image kind of describing it. So if you notice over here on the inside of that yeast cell, we only have two of those dye particles, but on the other side we have a whole lot of them. So in this, and but that arrow is pointing from the low side to the high side. That's the gradient. But if you also notice, there's ATP being used here. That's one of the biggest giveaways if I ask you to identify which one's active or passive is ATP being used. And then after that, make sure the arrow is pointing the right way. Is it going low to high or high to low? If this arrow was actually the opposite and it was pointing from the big side, from the outside where all those dots are, and the arrow was going inside of the cell to where there's only two, that would actually be an incorrect answer, an incorrect diagram for it, because that the arrow would be showing passive, but yet there would be ATP involved, and that would just be nonsense. So make sure that you don't get confused between what an active transport image looks like and what a passive transport image looks like. So the sodium potassium pump, basically all this is the transport of sodium and potassium against the gradient. And look at this. We're also using energy. ATP is being used in that image. If you look um, outside the cell, where you see that K plus, that's potassium. And if you follow that arrow is going inside of the cell, and that's where all those other Ks are. All those other potassium molecules are. All those other potassium ions, they're all being moved. They're being moved to an area where they're all at. Same thing with the sodium. It's being moved out. That's moving from a low concentration of potassium to a high concentration of potassium, just like the sodium is doing. Okay? So endocytosis, I'm pretty sure that you can understand what this term means. If you just break this down, endo would be to enter, E-N, enter, cytosis, that cyto means cell. So it's to move substances into the cell but against the concentration gradient. Remember, it's active transport. We're going against the gradient. So we're moving things into the cell against the gradient. There are two types here. We call those phagocytosis and pinocytosis. Another word you might see here, other than enter and into, and I have it right here beside phagocytosis, I talk about engulfing. So phagocytosis is whenever it's transporting a solid object into the cell. So whenever a cell engulfs something, Or we could be talking about an amoeba engulfing some type of bacterium in order to absorb the nutrients, or whatever the food particle is, in order to absorb the nutrients from it. Pinocytosis would be transporting fluid. So basically, it's fluid that has a bunch of dissolved particles in it, and it would be transporting that into the cell. Make sure that we know that endocytosis will only move things into the cell. Okay, and do know the difference between phago and pinocytosis. So this is an image. So if you were in class doing this, I would actually be having the image up on the board that I would be drawing on the whiteboard for you. So I had to make do with this. So I just did it on some post-it notes and then kind of drew some arrows. So by endocytosis, let's just kind of follow the pathway. So right here we have a cell. We have a particle outside the cell. And then over here we start seeing that particle kind of indenting into that cell. And then we come down to 3, it's starting to engulf more of it. And then by the time we get to the 4th image, we see that the cell is completely surrounding it. And if you notice, it has this double ring going around it. Well, the outside one, that's actually a part of that cell membrane. That's that, it has now its own phospholipid bilayer to it. And if this was an animal cell, the lysosome could go over there and break this down and digest it, do whatever it needs to do to it, because that's where those digestive enzymes are at. So exocytosis, if you look at that prefix there, exo means to exit, cyto, the cell. As substances move against the concentration gradient out, or to exit out of the cell. A good example of this is talking about that Golgi. Remember how we said it packaged things up and then it could send it around in the cell? Well, it could also send things outside of the cell. You could also look at calcium ions. They'll move this way as well. So this right here is talking about exocytosis. So right here we would have a particle inside of the cell, and each time we move to a new image, we start seeing that it's starting to move out of the cell, until we get all the way to the 4th image down here, where the particle has moved completely out of it. Just kind of follow the arrows, and then you see it move out of it. And that's it for active transport. So make sure that you can describe the concentration gradient for active transport, going from low to high against the gradient using energy. Energy must be involved. What molecule is needed for active transport? That's that ATP molecule, that energy molecule. Make sure you know the difference between phagocytosis and pino, just like you need to make sure you know the difference between endocytosis and exocytosis. Phago and pino both go with endocytosis, but do make sure you know the difference in it. And number 4 is like that image I showed you earlier on. Okay, and so that is it for this unit. Our next video, day 4's video, is just going to be a review over it. I'm just going to kind of recap through these review questions. Please make sure that you look at your slides. Don't forget you do have those quizzes that you can look back at and go over and kind of work through some of that information.